Have you ever wondered how your body knows to grow taller, produce melanin for your skin color, or even fight off a cold? The answer lies in a fundamental concept of biology called the Central Dogma. This powerful mental model not only explains how life functions at its core, but also provides insights that can be applied far beyond the realm of biology. Imagine you're building a house. You start with a detailed blueprint, which guides the construction process. The builders follow these instructions to create the physical structure. Now, what if I told you that your body operates in a similar way? The Central Dogma of Biology is essentially the blueprint of life, describing how genetic information is used to build and maintain living organisms. Just as a blueprint contains coded information that's translated into a physical structure, your DNA contains coded genetic information that's translated into the proteins that make up your body. This process is the foundation of all life as we know it, from the tiniest bacteria to the most complex mammals. At its core, the Central Dogma of Biology describes the flow of genetic information within a biological system. It can be summarized in a simple statement. DNA makes RNA, and RNA makes protein. But what does this really mean? Let's break it down step by step. DNA, deoxyribonucleic acid, is the storage molecule for genetic information. Think of it as a vast library containing all the instructions needed to build and maintain an organism. These instructions are written in a four-letter alphabet, A, T, C, and G, representing the four nucleotide bases that make up DNA. The first step in the Central Dogma is transcription. During this process, the information stored in DNA is copied into a molecule called RNA, ribonucleic acid. RNA is similar to DNA but is single-stranded and uses the base U, uracil, instead of T, thymine. Imagine transcription as making a working copy of the master blueprint. Instead of risking damage to the original plans by constantly referring to them, you create a more accessible copy that can be easily distributed and used. The second step is translation. Here, the information in the RNA is used to build proteins. Proteins are the workhorses of the cell, performing a vast array of functions from structural support to catalyzing chemical reactions. In this step, the RNA code is read in groups of 3 nucleotides called codons. Each codon corresponds to a specific amino acid, the building blocks of proteins. As the RNA is read, amino acids are strung together in the correct sequence to form a protein. Think of translation as the actual construction process. The RNA instructions are read by cellular machinery, ribosomes, which assemble the protein building using amino acids as the raw materials. The Central Dogma is crucial for several reasons. 1. Universal language. This process is fundamentally the same in all living organisms, from bacteria to humans. It represents a universal language of life. 2. Basis of heredity. It explains how genetic traits are passed from one generation to the next. 3. Source of variation. Mutations in DNA can lead to changes in RNA and proteins, driving evolution and biodiversity. 4. Foundation of molecular biology. Understanding the Central Dogma has paved the way for numerous scientific and medical advancements, from genetic engineering to personalized medicine. While the Central Dogma is a biological concept, its principles can be applied to various fields. Let's explore some examples. In IT, we can draw parallels between the Central Dogma and data processing. Source code, RNA, compiled code, protein, executable program. Just as DNA is transcribed to RNA and then translated to proteins, source code is compiled into an intermediate form and then translated into an executable program. The Central Dogma can be applied to organizational structure and communication. DNA, company vision and core values. RNA, strategic plans and policies. Protein, day-to-day operations and employee actions. The company's vision, DNA, is transcribed into strategic plans, RNA, which are then translated into actual operations and employee behaviors, proteins. In education, we can see a similar flow of information. DNA, curriculum and learning objectives. RNA, lesson plans and teaching materials. Protein, student knowledge and skills. The curriculum, DNA, is transcribed into specific lesson plans, RNA, which are then translated into actual student learning outcomes, proteins. Even in personal growth, we can apply this model. DNA, core values and life goals. RNA, action plans and habits. Protein, actual behaviors and achievements. Your core values and life goals, DNA, are transcribed into specific action plans and habits, RNA, which then translate into your actual behaviors and achievements, proteins. Let's consider a hypothetical scenario where a tech startup is struggling with innovation. How might we apply the Central Dogma model to address this issue? Company X is a software company that has been successful with its initial product but is now struggling to innovate and stay ahead of competitors. DNA, company vision and core values. Is the company's vision still relevant and inspiring? Do the core values encourage innovation and risk-taking? RNA, strategic plans and policies. Are the company's strategies aligned with its vision? Do policies support or hinder creative thinking and experimentation? Protein, day-to-day operations and employee actions. How do employees actually spend their time? Are there opportunities for brainstorming and developing new ideas? 1. Identify your DNA. In any system or project, start by clearly defining your fundamental principles, goals, or vision. This is your DNA, the core information that will guide everything else. 2. Create effective RNA. Develop clear, actionable plans or strategies that accurately reflect your core principles. Ensure that nothing gets lost in transcription between your vision and your plans. 3. Monitor your protein output. Regularly assess the end results or outputs of your system. Are they accurately reflecting your original intentions and plans? 4. Look for mutations. Be open to beneficial changes or innovations that may arise unexpectedly. Just as genetic mutations can lead to advantageous traits, unexpected variations in your plans or outputs might lead to positive developments. 5. Maintain information integrity. Establish systems to ensure that information flows accurately through each stage of your process, just as biological systems have mechanisms to ensure accurate DNA replication and protein synthesis. The central dogma of biology is more than just a biological principle. It's a powerful mental model for understanding information flow and transformation in various systems. By applying this model, we can 1. Analyze complex systems. Break down complicated processes into distinct stages of information transfer and transformation. 2. Identify bottlenecks. Pinpoint where information flow might be disrupted or altered in a system. 3. Ensure consistency. Check that the final outputs, proteins, align with the original intentions. 4. Foster innovation. Understand how small changes, mutations, can lead to significant innovations. 5. Improve communication. Recognize the importance of accurate information transfer at each stage of a process. The central dogma of biology offers us a fascinating glimpse into the fundamental processes of life. But more than that, it provides a versatile mental model that can be applied far beyond the realm of biology. By understanding how information flows from a core set of instructions, DNA, through an intermediate form, RNA, to final functional units, proteins, we can gain insights into everything from software development to organizational management. As you move forward, challenge yourself to apply this model to different aspects of your life or work. How can you ensure that your DNA, your core values, vision, or goals, is accurately transcribed and translated into real-world actions and results?